most of them are coming, and they are admitted in this venture. I'm very much grateful to the teachers of Vishnu Ayurveda College, and also PG students and house surgeons, and from all the students from the PG departments of Kerala. I welcome all. It's an honor for me to have such a function here. Dr. Lakshmi Chechi, she is only my sister, elder sister, and I'm very much happy that she is working in a US Tufts University that stands 37th in the world universities grading. So such a great university. She is doing the work of molecular biology. It is really we are blessed to have her, and I had the privilege to call her father for a ceremony for such type of class in the Kottakkal Ayurveda College, and due to some strike, he could not come. So at that time, I told we will call you once more, but we could not do that because he was no more. So at least in this, I could invite Dr. NVK Warrior's daughter, such a legend in Ayurveda. His daughter also is a legend. So we are blessed to have her here for a enlightening lecture in the research in Ayurveda. She has undergone very tough trials on these research works, and it is very much, I have heard some of the points she has told, and she will be enlightening you in what area we should, how we can do an Ayurvedic research, in what fashion you have to go through it, and some of the and Dr. NVK Warriors, he, as a legend of Ayurveda, that Ayurvedic knowledge also and passion also, Dr. Lakshmi Pulakkat has got it, and she is still now, though she is studied in the molecular biology, etc., she is very expert even in and trying very much to have research in Ayurvedic drugs and plants. And one of the products which Dr. NVK Warrior has introduced, the Sanjeevani Thailam, it is giving results for many cancer patients in Ayurvedishala. I don't say too much. I heartily welcome Dr. Lakshmi Pulakkat for such an auspicious function. Thank you for coming here. Chechi, you can start. Okay, thank you, Chechi.
Thank you for that kind introduction, Dr. Kiradamurthy. And actually, you don't need to thank me much, because for me, this is like homecoming. As some of you know, I grew up in Shoranur, and this is Shoranur College. It was not there when I was growing up. So I am so happy to see that an Ayurveda college, and now with a PG program, is existing in Shoranur. And it is my pleasure to do this talk and to meet with all of you. So if I can share, I will get started. Okay, can you see? Please unmute. And also, yeah. And also,
we can see.
Okay. This is a very informal talk. Okay. So please feel free to stop me anytime you have a question. Please unmute and ask. That will be the easiest way. So today, what Dr. Kiradamurthy asked me to do is to talk about research experiences in Ayurveda. So basically, I am a molecular biologist, as he said, and my look into the field is from the viewpoint of a cell and molecular biologist. But this is a necessary thing because, as Dr. Kiradamurthy said previously, this is something that I grew up with and I want to look into to get a better understanding of how these time-tested herbal preparations actually work. Because the biggest problem we face is the lack of acceptance of these drugs and these theories into the mainstream mainstream medicine. So, and especially in places like US, this approach is not particularly supported, although National Institute of Health, in fact, support alternative medicine, research into alternative medicine. So that's where we stand. And we do need a look at the three doshas in Ayurveda. How do they, and the medications and the treatment strategies based on these ideas actually provide benefits, but what we miss is how these benefits are functioning at a cellular and molecular level. And when you compare with the allopathic medicines, that is the advantage that is there. And that is where we need to take the research to make Ayurveda in the mainstream medicine. So this is my general thinking. So as you all know, and I don't need to tell you any of this, the meaning of Ayurveda is science of life, and it has an evolutionary history of over 2000 years of healthcare. It's a knowledge system that developed its methods by for validating both treatments and medicines and interventions. It has an impressive lineup of textbooks and teachers, as you all know, from, since 2000 years ago to now, constituting perhaps the most extensive medical literature in the world. And definitely there is a high potential for drug discovery breakthroughs, as herbs and formulations of Ayurveda are already being used in humans, and there is empirical knowledge about their clinical efficacy and safety. So the topics discussed today is to pick one instance, a current challenge for physicians of all medical systems, and then ask the question, based on our Ayurvedic knowledge, can we do something about this challenge? And I will come to what it is in a minute. And then I'm talking about the research methods, which is probably an important part of this talk, to give you a feel for how do we go from having a plant product to researching and trying to understand new insights into the mode of action and molecular targets of such widely used Ayurvedic herbs. So this is generally what I am going to talk about today. And as I said, if you have any questions, please unmute and ask. So I grew up listening to the constant conversations of these two Ayurvedic physicians, Ayurvedian N.V.K. Vadiyar and Ayurvedian P. Madhavikutty. And they had a Ayurveda pharmacy, Ayurveda pharmacy in Shoranur. And that's how I have learned whatever I know. And basically, I have had a understanding of looking with a holistic approach, even though I am working on a molecular and cellular biology field. So now let us look at one of the most challenging questions for all medical systems, and that is the multi-drug resistant bacterial infections by biofilm-forming opportunistic pathogens. What you see here is the worst of the 18 most alarming antibiotic-resistant threats identified by the Central for Disease Control. And among them, is particularly there are two molecules or two cells that two type of bacteria that I would like to take your attention to. One is this methicillin-resistant Staphylococcus aureus, and the other is the multi-drug resistant Pseudomonas aeruginosa. These bacteria are there in all over the everywhere in the mud, in the water, and we get exposed to them all the time. But because of our ability to to fight the bacterial infections, natural ability to fight bacterial infections because of our immune system, we do not generally develop the disease. However, what you see here is a patient who had to undergo foot amputation, and this is a diabetic patient. And as you know, diabetes is a major problem that causes an immunocompromised situation. And this is a kind of, most of the time, these leg amputations come in because of the severe infection by multi-drug resistant and biofilm forming opportunistic pathogens. And there is no effective methods. There's no effective antibiotic treatment for this kind of conditions. And that's why it usually end up into amputation. And this is still the story. And so that explains the need for better treatments. And this is where various medical systems have to come together, bridge the gap between them, and try to come up with better approaches. Similarly, this bacteria, Pseudomonas aeruginosa, which I will talk about, not only is a cause for wound infections, but is also a common cause of hospital-acquired pneumonia associated with the high mortality rates in patients with a compromised immune system. And once again, there are no effective antibiotic treatments. So what is really the problem? So the real issue is that normal bacteria that you see in water or mud that we often get onto our body would grow slowly, and usually our body can fight it off. But once they are capable of getting into the body and the body cannot fight it out, then they would attach, and they can do that on other surfaces as well. They would attach, and then they would form this stuff, what is called a biofilm. So these bacteria, which are outside and floating, free floating, can be attacked by various antibiotic treatments. However, if they become multi-drug resistant, then it is not that easy. They would be resistant to the antibiotics. Also, often low level of antibiotic treatments can induce this biofilm formation. The biofilm is a thick wrap around these bacteria that protect them, to put it very simply, that protect them from both antibodies of our body that can fight bacteria as well as antibiotics. So unless we can break the biofilm and get the bacteria free, they would not become susceptible to treatments. And that's one of the major issues. The second issue is that, as you can see here, the antibiotics, which is in yellow, may fail to penetrate beyond the surface layers of the biofilm. And so the bacteria inside is well protected. Some bacteria may differentiate into a protected phenotypic state, which are these. And then in zones of nutrient depletion or waste product accumulation, that is down here, antibiotic action may be antagonized. So for many reasons, these bacteria survive in the body, continue to infect, and make, whether it is lung infection or wound infection, skin and soft tissue infections, etc., make them really worse, leading to high mortality rate. And this is just to show you a little bit more in-depth and understanding of this particular bacteria. And I'm just taking this as an example, but I'll explain why soon. Why is it so difficult to treat? If it is in this format, which is the without the biofilm, it is not too difficult to treat, although it is resistant to many antibiotics. And if it is in this format, then antibiotics cannot get in. And that is what is shown in more clearly down here. The Pseudomonas aeruginosa biofilm cells, which are these like grayish cells. And you can try whatever you want, but it is not easy to break through this. But on the other hand, within this, other bacteria can get in, and bacteria get degraded. And instead of just P. aeruginosa or Pseudomonas aeruginosa here, you can have multibacterial colonies which share multi-drug resistance between them and overall become an extremely difficult infection to treat. So our research question was to do a pilot study to test whether any of the Ayurvedic herbs can stop infections by biofilm forming P. aeruginosa. In this case, what we wanted to use was a series of various Ayurvedic herbal approaches. And I will specifically talk about Tripharadi Churnam because it was the favorite treatment that my parents used for many, many conditions. So in this simple experiment, this was just a pilot experiment, we used Tripharadi Churnam and its ingredients. And I want to say Tripharadi here because you have the three specific ingredients of Triphala, and also this also contains licorice when you call it Tripharadi Churnam that Kottakkal Ayurveda makes, and the same thing that was my mother used to make. So then we have turmeric, black pepper, cinnamon, coriander, and cumin, all herbal components that are known to have antimicrobial effects. So here are the different components that we tried, and here is also Triphala itself. And so I think you are familiar with this Terminalia chebula. It's a haritaki, and then from the pictures you can see which one are there. Terminalia bellarica is, I don't know, it's a English Sanskrit name, but it's Tanika in Malayalam. And then this is, of course, licorice. And then Triphala is a combination of, and this is amla. And this is, of course, is a combination of all four. And then these are self-explanatory. This is turmeric, black pepper, cinnamon, coriander, and cumin. So this is what we basically did was to just take a 1 gram to 1 ml kind of dissolving. So this is like a thick paste kind of stuff. And that's what we originally used without doing anything to it as it is. And what we did then is the first step was to develop a biofilm producing bacteria, biofilm producing P. aeruginosa. Because in the lab, what we had was a biosafety level 1 P. aeruginosa that we can use without any complications, and that's called PaO1. And we could get this from American type culture collection. What you see here is a medium in which this PaO1 is grown. And this is a Petri plate, and it has a medium. And the reason why this is so blue in color is because P. aeruginosa produces pyocyanin, which is blue in color. When you see often pus with the greenish, bluish green color, you know it is a P. aeruginosa infection. So this is this color, and this is how they grow. And what we did is, for biofilm to form, you need specific surface. So it's a very simple approach. We put some pipette tips on this Petri dish and plated the plate with the bacterial cells and just let it grow. What happens is that bacteria grows all over the plate but however, some of it get onto this plastic surface. And then they adhere. And then we can see that they start developing a kind of a tinge, a reddish tinge. At this point, we take them and put onto new plates, and we let them grow. And then we continue to make them grow and repeat this process over and over and over. And what happens now is that we get cells on these pipettes that are not the original bacteria that does not produce biofilm, but the bacteria that actually produces biofilm. So this is what we did. And when I say mucoid colony, which means that the colony look like slimy because it has a biofilm on it. And so this is a biofilm forming bacteria. And usually people, you get biofilm forming bacteria either from wound infections or other kind of techniques. But in here, we used a very simple technique to get this bacteria, and then a single colony was selected and put on a new plate, and it grew well, and we get this, mucoid biofilm forming bacteria. And this is now called P. aeruginosa MG1. We checked and made sure that we are not working with some, some contaminant. And for that, we did the basic molecular microbiology test, because the Pseudomonas aeruginosa, when you put through testing for the presence of catalase and the presence of oxidase and presence of gram-positive or negative staining, the classic Pseudomonas aeruginosa is catalase positive, oxidase negative, and gram negative. And you like E. coli, which is also gram negative. So these features is the classic P. aeruginosa features, and we saw that they were also the same for the MG1 strain. So this confirmed that we are working with the P. aeruginosa strain, which is forming a biofilm. The next thing we did is to test whether all these different extracts from these different herbs, what will they do if we drop it, a tiny drop, on the bacterial Petri dishes where the bacteria is growing fully. And this is called the spot test. So what we have done is to go through this, and you can see that ignore the dark color. The dark color in the center is just because we are putting something like a thick brown colored paste for most of these herbs. But what you're really noticing is the cleared area, and it's more clear in here, over here and here, both you can see the extent of the cleared area where the bacterial growth is cleared off. And it's easier to see because this one is more dark blue. And what we noticed was that among this is Terminalia bellerica, and this is the licorice, and the other ingredients, and this is the amla. And compared to these ingredients, it was Terminalia chibula or haritaki and triphala that had the best effect. And so haritaki probably had the best effect, partly because Terminalia chibula is one of its ingredients. And at this point, it is what we call, this is like a highly concentrated solution, and what we have used. So then what we did is we did a 20-fold dilution of these extracts and asked the question whether we can still have the effect. And once again, as you can see, the Terminalia chibula gave the best effect in terms of, and that is more clear in here. So all these experiments were repeated several times, and all what I'm showing here is the examples of these Petri plates. So the next question, of course, was, we took all these different things like Terminalia chibula, for example, and a couple of Terminalia bellerica, etc., and checked their pH. to our surprise, the pH they were extremely acidic, and I think that is expected. And so the question obviously was that whether this nice positive effect that we are getting is because of the acidic nature of the herbal extract, or because of specific molecules, specific components that are part of this herbal extract. So the first thing we did is to spin it down or centrifuge it so that we get the solution, and which is here. And then we neutralized the pH, which is this one, is the N neutralized pH. And then we also passed it through a 0.1 micron filter to see, when we pass through such a filter, whether the effect is lost. And as you can see, even this one, you can see all of these better with the PaO1, and you can see that in all cases, the extent of effect was similar. In other words, we can neutralize the pH, we can pass through the filter so that the component that is causing the effect is not because of the acidity, and it is something that is smaller than 0.1 micron. So then we went and checked whether we could inhibit the growth of P. aeruginosa strains in liquid cultures, because up to this point we were using Petri plates. And this is in the liquid culture where we could actually follow the growth. And this is what you measure the optical density of the culture at 600 nanometer. And you can see that the basic cells were no treatment, grow nicely over the hours. However, when you treat with either 10% terminally achievable solution or 1% terminally achievable solution, the growth is completely gone. But in these cases, we have added the bacterial cells into the liquid culture and at the same time added the extracts. But most of the time, you get the infection where the bacteria is already grown. So then we wanted to confirm whether we can stop a already grown bacteria. And so in this experiment, we let the cells grow, and they both grew in the same way. And once they grew fairly well, with the optical density of about 0.6, at that point right here, we added the 5% terminally achievable extract. And immediately, where the terminally achievable extract was added, the growth came down. And then the growth was fully stopped in the next couple of hours, as long as we checked. There was no regrowth. So this was pretty good confirmation that the bacterial extract, the terminally achievable extract that is at a diluted stage can nicely destroy this bacteria. And one more point was that this particular extract at this point, what we are using is also pH neutralized and autoclaved to show that after autoclaving or putting at such high pressure, the antibacterial component was still functional. And then this is how we try to identify the lowest concentration of terminally achievable extract needed to inhibit the growth of P. aeruginosa. So here we used, in this case, we have the cells are just growing. We have not added anything to them, and they continue to grow. And then we started using varying concentrations of the terminally achievable. By the time we use 1%, the growth is pretty much fully inhibited. However, even at 0.125%, the half of the growth is inhibited. So we started seeing we started getting a better understanding of our autoclaved and neutralized terminally achievable extract, and are also passed through a 0.1 micron filter. So it is sterilized in multiple ways. And this particular terminally achievable extract, it can nicely inhibit bacterial growth. So these are all the various methods that we used. And so we continue to follow that. And the next question we had was that, how do we determine all of this work we are doing here? We have put on the plates and we have looked at, whether they can inhibit biofilm forming Pseudomonas aeruginosa MG1 and the non-biofilm forming PaO1. But we don't have a real measure of what is the minimum inhibitory concentration of this Terminalia tubula and how does it compare with other bacteria which it can inhibit and with other antibiotics. So to do this, we decided to use a very simple device called Calgary biofilm device. This is basically a 96 well plate that many people would be familiar with if you grow either mammalian cells or bacterial cells. But the difference of this plate is simply that it has got within each well a little stick or a peg on it. So the advantage is that you can grow the bacteria in here. Just like we put the pipette tips on a plate and let the bacteria adhere to the pipette tips. In the same way, in this case, it is done in a much smaller scale, so we can do a lot of experiments at the same time. And the bacteria will adhere to these little pegs inside each well. So what we can do is this Calgary biofilm device is basically a platform that generates 96 equivalent biofilms for the assay of antibiotic susceptibilities by the standard 96 well technology. So what we can do is that we can grow the bacteria for a few days and then wash off all the bacteria using some sterile saline solution. When you wash these wells multiple times, all the bacteria will be gone. Just like when you wash a wound, all the top surface bacteria will be gone. However, the bacteria that is in the biofilm, that is attached to the peg, will not go away. So to get that bacteria out, you have to do something like something like a sonication type technique to remove the bacteria. So what we do is we grow the bacteria and then add various drugs. And after adding the drugs, then we will do an initial testing to see how the drug has worked, which will provide us how it has killed the naturally existing bacteria, like the non-biofilm forming bacteria. And then we wash off all that stuff and then disrupt the biofilm and then ask the question whether the drug did anything to the bacteria within the biofilm. So this is how we do it. And so the explanation for this one is minimum inhibitory concentration. And just to explain, the relationship between MIC and zone of inhibition is that the MIC and zone diameter of inhibition are inversely correlated. The more susceptible the microorganism to the antimicrobial agent, the lower the MIC and larger the zone of inhibition, which is what you saw, what I showed you in those earlier photos, images, where if you add a Terminalia tubula, let me go back to one of these. Yeah, you can see this area which is cleared. So the larger the area that is cleared, the MIC will be the lower the number. So with this explanation, let me go to the experiment. So before I get to that, let me also introduce one more bacteria, and that is Staphylococcus aureus, which was one of the Central for Disease Control identified notorious biofilm-forming bacteria. It's an important agent of wound infections, boils, pustules, and other human skin and soft tissue infections. Many strains of Staphylococcus aureus are now resistant to penicillin and methicillin. These antibiotic-resistant strains have become a major source of nosocomial, in other words, hospital-acquired or community-acquired infections. And these are, again, what we call opportunistic pathogens, meaning that they are in the soil, they are in the water, they are everywhere, and we usually fight them off But if the body become immunocompromised because of surgery or age, including age, many conditions that can cause your immune system to be weak, and or cancer treatment, chemotherapy, etc. In such conditions, and also metabolic diseases such as obesity and diabetes, in such conditions, the infection by these bacteria can increase. So keeping this in mind, we asked the question, what is the comparison of the MIC to other MIC and biofilm eradication concentration? That's one thing I forgot to explain. So the other thing we look for is minimum biofilm eradication concentration, or MBEC. And this assay is used to determine the efficacy of an antibiotic, antimicrobial agent against biofilm. This is what I was explaining about the Calgary biofilm device. So having done that, we then went and checked what is the MIC and what is the MBEC of different well-known antibiotics, as well as our Terminalia cibula extract, and the how much material you need. So these ones are microgram per ml, all of these. And this is tetracycline is dissolved in ethanol, ciprofloxacin is in ethanol, and the erythromycin is in 0.1% hydrochloric acid. This is what we have used. And then Terminalia cibula extract, autoclaved and pH neutralized, and diluted to different levels. So what we found was that the P. aeruginosa PA01 is the non-biofilm forming bacteria, and the MG1 is the biofilm forming bacteria. And as you can see, when you come to the biofilm forming bacteria, we need high concentrations of tetracycline and to get some inhibition. If you use ciprofloxacin, then when you come to the biofilm inhibition, the dose that you need is much higher. When you come to the erythromycin, same story. When you come to the non-biofilm forming, the dose is not that high, but when you come to biofilm forming, the dose is extremely high. So this is going to be the case in whichever way it goes. But what is good here, especially, is that you can see that P. aeruginosa MG1, for that, the MIC and MBEC both are same numbers. So it's very well capable of inhibiting this biofilm forming MG1. Now we go to Staph aureus, and the Staph aureus, Terminalia cibula could inhibit much better. And once again, the MIC and the MBEC are very similar. And then you start doing co-culturing, so the two bacteria together. And now you can see that all of these ones can still inhibit, but at a slightly higher concentration. And none of them are capable, especially ciprofloxacin and azithromycin, are not capable of completely eradicating. We are looking at minimum inhibitory concentration. And here you can see that even if you use the combined cultures, when you look at the biofilm forming and the Staphylococcus aureus, we can get very similar effect as we got for the single ones. So in general, all of this data indicate that Terminalia cibula extract is powerful in treating these biofilm forming bacterial infections. So this leads us to the question, how does this work? And so this is the last slide that I have. So what we did to understand that is to take these bacteria and expose them to these minimum inhibitory concentrations, and then isolate the RNA from these, or ribonucleic acid from these bacteria, and then look at what is happening to the gene expression in these bacteria. So we would get something like 2,000 different genes that we analyze, And out of them, a handful of genes show significant difference in the way they express in the untreated, in the presence, and untreated, like no treatment and the treatment. Here, when I say presence, that just means that we can see the expression of the gene. However, in the untreated bacteria, the expression levels are quite higher, and the treated ones, it is when I say the fold change compared to the treated ones, this is the number of times these particular genes are expressed in in the bacteria. Or in other words, in the presence of treatment, the expression of these genes is substantially suppressed. So what are these genes that are substantially suppressed? And that is what is highlighted here particularly. All these genes are necessary and essential for the growth of the bacteria in a biofilm condition. Because if you think about it, when the bacteria is exposed in the water or air, it has easy access to oxygen. So it can breathe and grow normally. But if it is encapsulated in a biofilm, then the oxygen levels are extremely low. So under those conditions, the bacteria go into a different mode of growth. And in that condition, bacteria has to grow in anaerobic condition without oxygen, as well as it has to use anaerobic gene expression to utilize nitrite and nitrate in the medium to be able to grow. If that anaerobic gene expression is suppressed, then the bacteria cannot grow. And it is not easy to suppress the anaerobic gene expression. And what you are seeing here is that multiple genes involved in promoting the ability of the bacteria to grow under anaerobic conditions, not one gene, several genes, are severely suppressed. And if they are severely suppressed, the cell will die because it just cannot grow when there is no oxygen. And that seems to be the trick by which Terminalia tubula inhibit these bacteria. So here's a lot more work to do to see whether we can take this to clinic and what more can we do. But that is where we stand. So thank you so much for this opportunity. And please let me know if you have any questions.
Now the session is open for the questions. If anyone have any questions or doubts, please ask.
Hello, madam. This is Dr. Devan from Shalya department. Madam, I have a doubt that we used to have patients come with diabetic wound ulcer. So we know very difficult sometimes to treat the case if the patient is not responding to the normal what we are giving. We used to give local applications, especially Triphala Kashaya wash and Jatyadi Taila for the wound healing purpose. When our practice time, we used to observe that sometimes the medicines which we are giving is not that much effective to produce the result in the infective conditions. The wound shows delay in healing as well as we need the support of antibiotics. So whether my question is the formulations when we are using like the Taila or the Grida preparation or the Kashaya preparation, like how when we use it in practice, like how effective, like in cases of infections. Like as you have told, like there is a resistance where we have to cross the film and act the medicines So our preparations like Thaila and all, whether like and during the practice, I have observed that sometimes after clearing the wound becomes Shuddha Avastha, then we get good results. But we are giving during the rough times or like after debriding also, the wound shows delay in healing by applying our normal formulations.
Yeah, so that's a very good question, and that is generally the question that we all like. It doesn't matter which medical field you go, you deal with. So when you are talking about diabetic wound particularly, and actually my primary research in right now is in diabetic wound healing, in trying to come up with better ways to treat obesity and diabetes associated wounds that don't heal. So when you think about it, there are multiple issues. One is that because the patient is diabetic, and how bad is the diabetic condition? Because when that is higher, then you have a lot of sugar in the blood that can actually help the bacteria to grow on one hand, but at the same time, you also have what we call is, you don't have enough capillaries going there to the wound area, because when you are severely diabetic, you have a serious microcirculatory problem. Blood vessel capillaries are either damaged or the blood, they are occluded, something or other. So what happens is, if you want to treat such wounds, you need pretty much a multi-pronged approach. And what I am showing was simply that Terminalia tubular has the ability, and if you can extract it, you can use a diluted molecule. And this, as I said, this is a preliminary study, and that is pretty effective as much as of some other antibiotics. However, the problem remains that can you get your kashayam or xylem or whatever deep into the tissue? Because the problem is, it's not only just that you have a exposed biofilm, but also that there is bacterial invasion deep into the tissue. So the treatments often can remove when you do debridement, etc., the top surface, but you probably are not catching the bacteria that is already gone deep into the tissue. So, and any treatments that we give, the problem is in such kind of wounds, the penetration is not great, whether it is antibiotic ointments, antibiotic creams, etc., or whether it is Ayurvedic medications. My thinking is Terminalia tubular component that is causing the antimicrobial effect is very similar or maybe even better compared to some of the antibiotics. However, it has to reach deep into the tissue. So this is part of the reason why I'm doing the study, because not this particular one, but interested in the diabetic wound. Because one of the things we propose in our lab is to try to inhibit the wound by approach that involves changing the gene expression in mammalian cells so that the cells have the ability to fight. But to do that, as well as provide antibiotics extremely deep into the tissue. And we use a device that is called droplet micromist technology device to do that, to penetrate deep into the tissue. So I would think that while the treatment that you are saying is effective in some cases and not effective in other cases, is partly because it's not penetrating deep into the tissue to kill the bacteria that is inside. And then in a day or two, those bacteria that are still there can grow and come back, even if the top surface is cleaned. Does that make sense?
Yes, madam.
Is there any other questions?
Well, feel free to ask. There's no, anything. It's all right.
Hello, madam.
Hello.
Can you hear me?
Yeah.
I'm Dr. Vinita.
Mhm.
Madam, you just wanted to say that now with this experiment, the direct contact of Terminalia tabula extract had given the effect of uh, resisting the biofilm formation, right?
Mhm.
Then I just want to add an experience. Years
Mhm.
back, we had treated a case with a Triphala Mashi. Actually, I have prepared that for a as an anjana in eye treatment.
Mhm.
But as the quantity was so excess because we were preparing it the first time, and the sticky nature of the Triphala is the tannins and all are very high, and that quantity outrated, and that quantity later we used in a case of this wound. So I think this Triphala Mashi preparation has got a more sticky nature, and it can more adhere to the wounded surface, and it was really effective.
Yeah, that sounds good, right? That's a positive thing for obviously. And I think that, so one thing I would say is when you are making that way, you can use, so you got the wound healing properly in this case?
Yes, madam. Yes, madam.
Mm-hmm. So that's what I'm saying. I got quite interested in Triphala because I do remember a case when I was a small child, but my parents treated, and this is a friend, a young lady who got all over the body boils. At the time, I did not know what it was, but now when I think about it, I believe it may have been Staph aureus infection, actually. And they didn't know what was happening, but it was the whole body, both back and front, which made it extremely difficult. And obviously, people were afraid there was high fever, etc. That goes with it. And also, people are afraid whether the young lady will be, disfigured in some way. But the treatment, my parents went and saw this girl, and the treatment they gave was one of the treatments they gave was Triphala and Triphala powder and Triphala, as you said, Triphala Kashayam to use all over the body. And in this case, the infection was not deep and the patient recovered without any major, scarring in the body. So I do know this is something that stuck with me from that time on, and that's why I selected Triphala as the primary molecule thing in here, and just wanted to test other herbs. How do they compare with Triphala? So the important point is that you don't need an extremely concentrated Triphala to make it work. It looks like, So, but however, it has to come in contact is the important point. And if the bacteria get trapped deep into the tissue, then we need a delivery system that will help this to deliver deep into the tissue, which might actually prevent the problem or, treat the wound much better. Anything else?
Anybody asking questions? Techi, you are telling about the experience of Sanjeevani Taila. Can you explain it some?
Sure. Yeah, that's a completely different thing. So Sanjeevani Thailam, this is something which I did, I think, in 1996, 97, those years, actually. So basically, Sanjeevani Thailam at that time was used by P.K. Warrior, late P.K. Warrior, and my father for cancer treatment. And it was really an experiment. So this is one of the advantages compared to in this, what I just showed you, the experiment, the set of experiments, we started with Triphala, then went into each component and then went into how to identify what is the target of this drug in terms of genes. This is the same approach I used there too. Father was very interested in understanding why and how Sanjeevani Thailam is inhibiting cancer growth. So the story, anecdotal stories within the family was that one of my aunt had a uterus cancer, if I remember correctly, and that was uterus had to be removed, and then it metastasized to breast. So it's my father's sister, and so she basically said she didn't want to go through the whole chemotherapy and all that, and if there is some other way, we'll do it. Otherwise, they gave her a 6-month time. And that's where what probably inspired father to seriously look into this. And they came up with a treatment which primarily involved Sanjeevani Thailam usage. And it's the external usage. And then there were other internal medications. The good news is that she lived for about, she passed away in, I think in 2006 or 7. And this she was diagnosed of the disease in, I think, 1990, probably. So obviously, the metastasized cancer in this case was somehow inhibited. And now there was no more growth. It's not, the tumor was still there. It's not like tumor disappeared, but it didn't reach to a point of death. So that was one that really took my interest into it, and father wanted to really know how does this work so that other people can use. So I got the Sanjeevani Thailam, and this is made of Nilapala in Malayalam. But when you say Nilapala, it has many different, it is not just one plant in that group. But particularly what they used was Euphorbia hirta, I think, and what they were using, and also one more variety of Euphorbia. And they usually, in the Kottakkal Ayurveda preparation, it is usually a combination. So I asked my mother to make each of them separately and make a very pure herbal product, which she did. And I then used that on breast cancer cells, 3 different types of breast cancer cells. One of them was estrogen positive, and the other one was triple negative, and one was herb B2, herb positive. These were the 3 kinds on which we attempted. So it was a very simple treatment. You grow the cells adhere because the cells have to adhere for them to grow in all these 3 cases. And what we did is we put a little bit of, we used this oil, like kind of put on top of them in the medium. And what happened, and as a control, because Sanjeevani Thailam is made in coconut oil was used as a control because I wanted to make sure that whatever effect I am seeing is not just because you put some oil in there and that oil has some effect. Interestingly, in all 3 cases, we found that the cells detached and cells floated. And then we took these floated cells, washed them, and tried to reattach, and they did not reattach. So that was a very simple thing. So then obviously the question was, how does it work? So I did essentially the same analysis, which is the transcriptomal analysis. In the same way we identified for the Terminalia tubula extract inhibit Pseudomonas via inhibiting its anaerobic growth. In the same way, we did an analysis to identify how does Sanjeevani Thailam stop the cancer cell growth. Because if it is stopping the cancer cell growth by, similar to our various chemotherapeutic agents, by simply inhibiting the cell proliferation and killing the cells, then we do have a problem. Because such kind of treatments are actually not good. Like, they are dangerous for the heart because almost all chemotherapy approaches that we use currently we say that people escape from the cancer, but then die of heart disease because they cause cardiac problem. So if you want something that will inhibit cancer cells rather than killing the cancer cells, killing the cells, that instead of that approach, you want something that stop their growth. Because if a small tumor is there in the tissue, that small tumor is not going to kill you. The death comes because the tumor grows larger and tumor spreads, and then it would go to all vital organs and then inhibit the vital organ function. That's from where the death comes. But on the other hand, if you have a tumor in the breast and that, or any other kind of tumor in soft tissues, if you can just stop it from any more growing, then you don't have to worry about cardiac problems and other kind of stuff. Because cardiac problem happen because of the killing nature of the various chemotherapeutic agents, as well as radiation therapy, etc. So when we analyzed, that's what we found in this case. We found that a series of genes that are involved in making the cells grow are stopped. And as a result, these cells, usually they will grow at a much faster rate, but they are arrested at what we call the G1 phase. So that's in the cell cycle. That's the story of that.
Thank you, madam. Thank you very much. It was very informative. Anish,
Sir.
you can have, if there are no other doubts, we can.
Okay, sir. Okay, sir. We will wind up.
Thank you.
Yeah.
Thank you for the opportunity.
Thanksgiving is there. He will tell you.
Thank you, Lakshmi Madam, for such a very insightful session. Now I invite Dr. Sridhe, first year PG scholar from Department of Panchakarma, Vishnu Ayurveda College, for a vote of thanks.
Good evening, everyone. On behalf of the PG scholars at Vishnu Ayurveda College, I would like to express our sincere gratitude to Dr. Lakshmi Pulakkat for showing her invaluable insights and experiences in Ayurvedic research. Your presentation was particularly enlightening, and it has shed light on the exciting possibilities within this field. We also extend our heartfelt thanks to our esteemed principal, Dr. Kiradamurthy sir, for providing us with this valuable opportunity to learn from such a distinguished scholar. A special thanks to all the participants who actively participated in the seminar. Thank you once again, Dr. Lakshmi ma'am, for your time and expertise. We are all grateful for this opportunity to learn from such a distinguished scholar. Thank you, ma'am.
Thank you.
Thank you, Chechi. Thank you. Thank you very much.
Bye. Bye.
Okay. Okay.